So good morning everyone on my behalf as well. So my name is Joanna Lempiainen and I am a postdoc in Ben Garcia's lab at the Washington University in St. Louis. So I will talk about a project in which we have used the Covaris for our sample preps. So the Garcia lab already has a couple of publications related to this project, and we are kind of continuing from there. So we are interested in the epigenetic mechanisms in malignant peripheral nerve sheath tumor, MPNST. So I will tell you a little bit about the background of this condition. So this is actually a sarcoma that forms in the connective tissues that surround the nerves, and it is often associated with a genetic condition called neurofibromatosis type 1, NF1, that normally forms these benign tumors along nerves. However, about 10% of these patients actually develop MPNST, which is a rare, aggressive, tends to metastasize, and most patients die within 5 years of diagnosis. And it is also found in children, but most cases occur between 20 to 50 years of age. And resection and chemotherapy are the current treatments, so there really aren't any alternative treatments. So there is a need to kind of understand this cancer better so that we could perhaps have alternative treatments for it. So in addition to these NF1 mutations, there has to be these kind of secondary mutations that drive the formation of the MPNST. And most often these mutations occur at the CDKN2A and P53 genes, but also at subunits of the PRC2 complex. And this protein complex actually binds to chromatin to repress expression of genes, so it deposits this H3K27 trimethylation. And in MPNST, the SAS12 and EED subunits are often mutated, and these mutations often co-occur with the CDKN2A and P53 mutations as well. And so there are some cases where the functionality of this complex is still retained, and so these cases have more hope. So the PRC2 loss basically predicts poor survival, and the loss of this Histone PTM can in some instances also be used to help diagnose or distinguish MPNST from other similar cancer types. So one of the previous papers I wanted to mention is from a previous postdoc, John Wojcik, in the Garcia lab. So he basically, his work ended up being published in 2019 in Cancer Research, and the aim of this project was to kind of identify how does the PRC2 loss promote sarcoma progression, or kind of try to understand some of these mechanisms. So he had these tumor samples from both PRC loss and PRC2 retained tumors, and so these were the formaldehyde-fixed paraffin-embedded tumor samples that he was using, and he used quantitative proteomics to characterize the total proteome and Histone PTMs from these tumors. And one of the main findings that he basically found was that antigen presentation proteins are significantly downregulated in these PRC2 loss cases, which would kind of partially explain why these PRC2 loss cases are more, worse than the retained one. And on the other hand, we see a loss of the K27 trimethylation, which you kind of expect for having the PRC2 complex functionality being lost anyways. But we also, interestingly, see upregulation of several different Histone PTMs. So you kind of have these global chains in the epigenetic landscape of the tumor, if you may. But the previous work focused only on the SAS12 mutants of the MPNST, whereas in reality there are also some cases where the EED subunit is mutated. So we are interested in looking at the difference between these two mutants. So whether the changes in the proteome and histone PTM profiles are similar in these mutants. Could there be common pathways that could be targeted in these mutant tumors, and perhaps more ambitiously, could there be epigenetic drug combinations that we could use to combat the more aggressive PRC2 loss MPNST? Then the other paper that I wanted to mention is also from John Wuschig. So this was done in collaboration with the Garcia lab, and it was published in 2020 in the Journal of Proteome Research. And the kind of point of this paper was to compare different ways of extracting protein from FFPE samples, and the positive control, or the kind of comparison, is flash frozen tissue, which was processed by probe sonication and S-trap. And so the take-home message from this paper is that one of these extraction methods work the best, and in this method we have direct extraction is direct solubilization into SDS, then we have ultrasonication with the Covaris, and then recovery using S-trap. And this combination or method was named HyperSol, so high yield protein extraction and recovery by direct solubilization. And at the time, the ultrasonication was done with the S220. So about the current project, so we are collaborating with another lab at the WashU, and they have experience doing these PDX mouse experiments. So they are planning to do a immune checkpoint inhibition combined with different epigenetic drugs, and then they will send us the tumor samples, and we can then analyze the tumor proteome and histone PTMs from these samples. So they gave us this kind of small set of test samples, and one of the purposes of this trial experiment was to kind of establish our protocol, since the lab had moved from UPenn to WashU, and the previous people had left and new people arrived. So then one of the questions was that can we get everything to work that we would need to work when we get those kind of the actual set of the samples. So this was a trial set of samples where we have FFPE and flash frozen MPNST tumors, and they are either from human or then the PDX mouse. And one of the things that I first wanted to test was the HyperSol. So I'm actually doing the full protein extraction from the FFPE using the HyperSol published protocol. But then we are kind of adding more into that. So we are also characterizing the histone PTMs and kind of additionally the RNA modifications. There, then in parallel, we did the frozen samples. So here I'm using a bead beater, and then we have a probe sonicator. So I guess this is kind of the classical way of processing the frozen samples. And then I am using RNA binding columns, where the RNA basically binds to the column and the proteins can be precipitated from the flow through. So we get all the RNA or all the proteins from one sample. And then the end kind of steps afterwards are the same as for the FFPE workflow. So one of the kind of benefits, one more thing I wanted to add. So one of the nice benefits for using the Covaris that I realized when I was doing the HyperSol protocol, that you can actually add the punches, the FFPE punches directly into the SDS buffer and then just sonicate them to homogenize. So you actually don't have to use a micropestle or anything to smash the punches. The sonication actually homogenizes them so well that you can literally just put those punches into the Covaris tubes and the instrument will take care of everything else. So that was really something I was surprised about. So moving on to the proteome, for the analysis we used this Zyx Xenotop 7600, injected 200 nanograms of protein, used a 45-minute microflow LC gradient for the swath analysis, and then for the data analysis we used the Spectronaut using a hybrid library. And here I have kind of the zoom in of the HyperSol workflow or the beginning of the workflow. So there are actually two sonication steps that are being used. So we have 5 minutes and 6 minutes sonication, and then we have two heating steps, 1 hour heating steps in between. And we have the E220 on our campus that we can use for the sample preps. And here I'm just listing the settings that I have used. I think these are from the paper, so there wasn't anything different here. And then we are using the microtubes that have the 130 microliter volume. So one of the things I was interested in was that whether increasing the sonication time, will that make any difference in extracting the protein? So is this already enough, or would increasing sonication time improve anything? So I basically tested by doing the 5-minute published sonication time for the first step, but then I also used a 10-minute sonication. But it seems that overall I'm getting more than 3,500 proteins in both cases, and the proteomes seem to correlate in these two samples really well. So my end conclusion from this was that I'm actually already achieving maximum extraction from FFPE by using the published protocol. So there doesn't seem to be any need to increase the sonication anymore. And the protein group quantities seem to be very similar in these two conditions. Another thing that I compared was the protein numbers between the FFPE and the frozen tissue. So I have the human tumor and then I have the PDX tumor. And the end conclusion from these is quite similar. So they seem to somewhat correlate, and the protein group quantities the averages seem to be similar. And overall, it seems that the FFPE samples seem to be processed very consistently. So my kind of conclusion from this was that the FFPE sample prep worked really well compared to the frozen one, and this could be something we could really use in the future as well. And for the Histone PTMs, so the Histone PTM analysis is kind of a speciality of the Garcia lab. So we have a long history doing that and several publications related to that. So I have basically done, or I've loaded the total cell lysates into a gel and Kumasi stained the gel, and then you can cut these histone bands out of the gel, and we have a published protocol for doing in gel propionylation and trypsinization of these samples. So here I'm just showing some selected Histone PTMs and their relative abundance, and here I'm showing all the PTMs we identified from one peptide at the H3 tail. So you have all these single modifications, we have the unmodified peptide, then we have all these different single modifications, and then also all the combinations. So there are actually, it's actually a huge amount of data you get from one sample. And from this one sample, we could identify more than 100 different histone PTMs or their combinations. So it's actually a huge amount of data you can get from one sample. The RNA modification field, I suppose that's kind of new. So we used our newest Ascent instrument for this, and we were able to identify many different RNA modifications from both the Covaris prepped FFPE and also from frozen tissue samples. And it is thought that some of these modifications can also participate into cancer progression. So this field is kind of new, and I think this is very interesting. So we can also identify RNA modifications from these samples. Another thing that I wanted to mention is that since we are interested in the epigenetics of the MPNST and different histone PTMs, we are also planning to do ChIP-seq of these samples. So we can actually use the E220 to do ChIP-seq in our project, and here I'm just showing preliminary optimization or a test using different sonication times with MPNST cell lines. So we can use the same instrument and, funnily enough, the same tubes that I use for the FFPE extraction. So I don't even have to get new tubes to do the ChIP-seq in this project. I can actually use the same tubes, consumables that we already have, and the settings are almost the same, except temperatures. I'm going a little bit lower with these cell lines since these are not in the SDS. So these are in a different buffer. So to summarize, majority of the MPNSTs are driven by these SAS12 and EED mutations that lead to the PRC2 loss and major epigenetic changes. And in our hands, the FFPE proteome that we prepped with the E220 seemed to correlate quite well with the frozen tissue, so we were happy with that, so that we can continue using the E220. And interestingly, we can also get the histone PTM and RNA modification data from these same samples, so we can actually get a huge amount of data from one sample, which is really, I think, a huge bonus. And lastly, I would like to mention that I'm giving a poster today, so if you are interested in talking more or have any questions or suggestions, I would be really glad to talk. So my poster number is 119. And finally, I would like to thank our lab, our collaborators, our funding, and previous member John for his work, and thank you for listening.